pretty good quorum.
Okay.
So I'll introduce Pat virtually. Pat earned her PhD at BU in biology, and she did her postdoc at Wilmer. And then she rose up the ranks at Harvard while researching retinal vascular development and growth. She's the Charles Scapins Professor of Ophthalmology and Pathology. She's the Vice Chair of Basic Research at Harvard in Ophthalmology. She's the Director of the Scapins Eye Research Institute. She's won many, many awards, including Arvo's Kogan Award and Proctor Award. She's been the primary mentor for over 15 graduate students and 40 postdocs. She has 167 publications and contributed significantly to the development of anti-VEGF drugs, which are now the mainstay for wet macular degeneration and diabetic retinopathy. And in addition to all these impressive things, she taught me something important about moderating a session. So we were co-moderators at a session at Arvo, and there was a questioner who kept going on and on and on and got into a heated discussion with the speaker. And this was my first time moderating, and I didn't know what to do. But Pat doesn't put up with any nonsense. And she just said, okay, guys, you can continue this discussion at the bar. And that shut them down immediately. They were satisfied with that. And I have to say, I've used that line a couple of times now when moderating sessions. So thank you for that. And thank you for teaching us so much about retinal vasculature. And now Pat's gonna speak about a novel role for the endothelial glycocalyx in angiogenesis.
Okay, thanks, Josh. So I don't know, I guess it's, since people are muted, it's hard to have people just yell questions out, but maybe if you put them in the chat and there's something compelling, Josh, you can monitor the chat and let me know, because I don't want to proceed if there's like a question that somebody wants to ask. I was told this should be around 40 minutes, so I tried to cut some stuff out. So I may refer to some I'm not showing, but I have the data if you want to see it. This is my new hobby. These are my bees, which are hopefully going to survive the Northeast winter, even if I don't. So I'm going to talk about this molecule. We did not discover this molecule. This molecule was actually out there, but I'll give you a couple of slides about how we came across it. It's one of those fishing expeditions that study section always gives you grief about. Actually, those things usually do turn something up. So I don't know why people don't want to admit that. So this is a molecule called Endomucin. By the standards of people who study mucins, like the ocular surface people or people who look at the reproductive tract, it's really quite a small mucin. The mucins that the ocular surface people work on are in the millions. This is 150 KD once glycosylated, but it qualifies because it has this extended extracellular domain that's O-glycosylated, and that O-glycosylation manages to keep it upright and rigid, and that's a characteristic of the mucins. It has a transmembrane domain and some potential phosphorylation sites, which we really haven't figured out yet. Interestingly, they're very highly conserved, yet we haven't found a function for them, but the high conservation is making me want to continue to look at that. So how did we actually end up finding this molecule? If you search this molecule, you would find a lot of papers because basically people use it as a marker for the endothelium. So it's a really good marker. The way we found it, this is work from Eric Ng, who was a graduate student in the lab many years ago. He's actually back at Scapins now as an assistant professor. And we were in the VEGF business, as you probably know. And we had mouse embryonic stem cells. And we could make cystic embroid bodies. I don't know how many people are familiar with that technology, but if you take embroid stem cells and you force them to adhere to one another, and you can do that by putting them in a hanging drop, they'll actually form embroid bodies, meaning various tissues will actually form. You can, you know, if you do it spontaneously, you can see heart, you can see beating tissue, you can see contracting tissue, you can push it in one direction or another. But if you actually then take these little balls that become actually visual to the eye and let them sit on plastic and then stain them here for an endothelial cell marker, you can see that they have what you might want to call a vasculature. And that would be vasculogenesis because it's de novo. There's no pre-existing vasculature there. And then you could see actually here that we cut sections and this lumen-like structures. So at that point, we were very interested in how tubes form, because even though we knew about VEGF and proliferation and migration, nobody really knew much about the actual mechanics of how you form a lumen. So what we found is in the null embroid bodies, this shows you another picture. This is just stained for, again, for CD31 PCAM. This is wild type embryonic bodies that have VEGF, make this, you know, nice tube-like structures. But if you make the embryonic bodies from VEGF-null ES cells, you get endothelium. So they're still staining for CD31, but they don't make any tubes. So we thought this might be a great model to find molecules involved in tube formation. And this was a long time ago. You can see from the reference, I can't see it here. I think it's 2004. So the technologies were not as vast as they are today. Nobody was doing much fancy arraying, but we did one array at the time. And I gotta get this to go. And I'm just showing you the very top 3 hits, comparing what was basically missing from VEGF null. So what was missing from VEGF null embroid bodies, which remember don't make tubes, was calveolin, endomucin, and HEY1. HEY1's a transcription factor downstream of Notch. Calveolin had sort of been already shown to have some role in tube formation, although it's not 100% worked out what's going on there. That calveolin knockouts have altered angiogenesis. So we picked up endomucin, and we only started working on this again many years later. We did a few things at the time. It didn't go anywhere. There weren't reagents. So we picked this up recently and have 2 stories that we've developed about this. I'm going to talk about one of them right now. There's a story about this molecule that's related to inflammation, which I can summarize if somebody has an interest in that area. But this is just a section of a mouse pancreas showing this interesting specificity of the staining. It stains veins, so you can see this collapsed thin-walled vessel, and capillary profiles, but it doesn't stain most arterial endothelium. So you can tell this is an artery by the thicker medial layer and by the fact that the intima is thrown into these sort of waves. And there are some arterial endothelium that express it, but not most. And we can go into that if you're interested. So interesting expression pattern, capillaries and veins, but not arteries. Also, it's apically localized. So this is a section. These are through skin, human skin actually, and you can see this is the endomucin on the apical surface. Here's the outside adventitial layer, and again another section showing apical localization. All of this actually led us to the first study, which got us to show that there's a role for endomucin in preventing leukocyte adhesion under resting conditions. And that's, you know, it's, again, a story that I can tell you about if you have an interest. I thought we're focusing more on the angiogenesis part right now. So I'm going to talk about that, and very happy to have input because it's not what we expected. So starting to work on this, I actually thought I was going to be but working on a project that wasn't VEGF. I just had time for a change. I thought this is a good thing. So where is endomucin? This is the glycocalyx. So the glycocalyx is actually meaning, glyco meaning sugar and calyx meaning coating. So it's the sugar coating basically of the endothelium. There's a reasonable literature about the glycocalyx. It's quite descriptive mostly. It varies throughout the vascular system. If you go from sort of the venous side to the arterial side, this is stained with Alcian blue and gets you to see this sort of fuzzy arrangement, but it's quite thick. It's highly charged, consists of, sorry, I keep jumping on back and forth, proteoglycans, glycoproteins, some glycolipids. And, again, a big significant structure. Has a number of functions. Again, it's got obviously permeability and barrier functions just based on the charge. So it can exclude molecules simply based on charge, but also probably has some steric capacity. It's been shown to be a mechanotransducer, has a role in anticoagulation. Obviously, there's probably heparin sulfate molecules on there. Anti-inflammatory, again, is the work that I told you that we've worked on. It does function in organizing cell surface receptors and does participate in intercellular communication because it does bind growth factors. A lot of growth factors that you think about, PDGF, VEGF, FGFs, they all bind heparin sulfate, which is part of the glycocalyx, the cell surface. And interestingly, its loss or modification is associated with a number of diseases. There's a huge literature about sepsis, again, very descriptive about changes in the glycocalyx that you see with sepsis, but also increasing literature about diabetes, atherosclerosis, and hypertension. So obviously an interesting place to look, not just from the viewpoint of biology, but what's the clinical significance of this structure, if you will. So reminding you back what we saw again and how we ended up going in this direction. This is a flat mount of a embroidered body stained for CD31 PCAM. This is the wild type and it stains nicely, co-localizes with endomucin. And then you make it the embroidered body from a VEGF-null and you have endothelium for sure, but there's no tube structures. So following the angiogenesis story. So simple hypothesis seems safe enough. Endomucin plays a role in vascular morphogenesis. So this is very sensitive. So this is work. We had antibodies to this. Some were commercially available.
I think something is frozen.
Yeah, Pat's internet is frozen. I'll tell her as soon as she comes back. That's a nice stop action photo of her there with the gesture. Hi, Pat. Can you hear me?
Yeah, I did. I just got lost, right?
Yeah, we lost you right when you started with this slide on expression of endomucin.
That's fine. So hopefully I won't get lost again. It says I have good service. So this is just, did you hear me say that the mouse is a good model for vascular development?
Just right at that moment is when you froze.
Okay, so hopefully it won't happen again. So the mouse retina gets vascularized postnatally. At P0, there's nothing. This is P3. You can see here right now, which now my pointer is not working. Okay, so why is my pointer not working? Oh, bummer. Oh, there it is. Okay, so this is the extent of the very primitive vascular network that you have at P3. The retina vascularizes from the optic nerve outward towards the periphery. You can see beautiful total staining for endomucin and the merge of PCAM. This in this case, not PCAM, it's isolectin that stains for the endothelium. And just 4 days later, you can see that this plexus has remodeled into a true arborizing network. These are arteries, veins, and these are the intermediate capillaries. And already by this time, the expression in the artery is gone. So endomucin expression is already gone at this point, and it's now restricted to veins in most of the capillaries. So very interesting. We know that there's actually have started to look at this, and there's a role for blood flow in the regulation of this molecule. So, and interestingly here, if you look at the very edge, so this is way out at the edge at the periphery, you can see that at the growing tips, the endomucin is even more highly expressed. So you can see how much more orange this is versus the backside. So this is where the migration and extension of the front is responding to local VEGF concentrations for vascularization. So we did some work. This is published, so I won't go into a ton of detail, but since nobody can read every paper or any papers hardly, I will talk about what we did. We're actually making knockout animals now. We finally have them, but in the beginning, when this work was done, we didn't. So we used siRNA. This molecule knocks down really nicely with Si. So this is preparation for in vivo studies. And this is just the background that we did, the work that we did. So Cindy did work to show what concentration you needed to use. Now, what I want to point out here is when you do an intravitreal injection with Si, we're probably only affecting about a third of the whole retina. So we're not, the Si is not going to reach all of the retinal vasculature, which is why when we look at what kind of knockdown we get, we don't see total knockdown because there's a good part of the retina that's not even affected, and I'll show you that. So this is, you'll see that the area that where we inject and make a little tattoo, we get actually quite good knockdown. And then we just looked at the time course, you know, how long does it stay knocked down? 42, 72 hours looks good. And so then she did an experiment where she injected this into, I think it was a P3 mouse or P4 mouse. I'll have to look again. And I'm so sorry to be doing that. I have to stop touching things. I'm going to just do this. You can see that when we put in the SI control, you have good isolectin stain and good endomucin. And when we put in SI endomucin in this, particularly in this area where the SI reaches, you have still good vessels, although you can already start to see some difference because you can see that these intercapillary spaces are bigger and the endomucin is gone from this area. And then if you bring your attention all the way out here, this is the flat mount of the whole retina, but if you bring your attention to the edge where this is going on, you can actually see pretty significant differences that in the SI control, you have tip cells are an area that people have been very interested in angiogenesis, like what's going on in the tip cell, how do they recognize VEGF, how do they know when to stop, how do they know they're migrating and not proliferating, et cetera. But if you look at the difference here, you see that these tip cells have very blunted surfaces and are missing these filopodia, which are a very important part of the whole sort of chemokinetic movement. And then we actually quantified this and we could show that the vascularized area was reduced. And again, the baseline is very high here because we're counting the whole retina, but only maybe 30% of the retina was actually influenced by the SI. Same thing, you reduce branch points, it's a way of measuring density, and then you reduce filopodia, which I showed you. So we had this observation that we could interfere with angiogenesis, but we had zero idea of what the mechanism was. So we went back in culture again, and for these studies, we're using human retinal endothelial cells, so primary retinal, not a line, primaries. So they're primary cells, and we're going to just show that we get really very excellent knockdown. So this is just what's happening with RNA expression at 24 and 48 hours. And then the protein is almost totally knocked down here. You can see that you get very excellent knockdown. We're also going to do sort of gain of function studies. So we made an adenovirus that overexpressed endomucin. Now the adenovirus actually overexpresses mouse, which will be relevant because it's a way for us to distinguish mouse from the endogenous human that the obvious human retinal endothelial cells make. So you see you get very significant levels of expression. You probably about double it up because you can see this is what baseline human is, and now we're overexpressing mouse. So what I'm going to show you in the next several slides is, look at my phone, okay, is assays that people generally agree are sort of components of angiogenesis. So we looked at proliferation, migration, and tube formation. And the results for each of those are pretty similar. So I'll spend the most time talking about the migration, and then you'll see that the others parallel it. So this migration assay is called a scratch assay. And all you do is plate the retinal endothelial cells, make a scratch, and then allow them to migrate. with VEGF. So if you add VEGF, so this is where the scratch used to be. Okay, you knock down, this is, you have to have the control, SI control, and these are the cells that have migrated in response to VEGF. They're just healing a wound. It's a measure. There's many migration assays. We just chose this one. If you knock down endomucin, though, you can just see visually that there's already less migration, and we quantify that here. On the flip side, and this I didn't really think would work, I figured that the cells were already migrating as fast as they could. I wouldn't have expected that we could have actually increased it. But again, this is we're overexpressing endomucin or a GFP control in response to VEGF. And again, this is how much migration you get. Looks about like this. But if you overexpress endomucin, you actually get increased migration. So endomucin, the lack of endomucin is preventing the cells from responding to VEGF. And the overexpression of endomucin is actually accelerating or increasing the response to VEGF. So I wasn't expecting to have a VEGF project with endomucin, but I do. We saw the same kind of a thing looking at proliferation. This is just looking at absolute cell number. And if you knock down endomucin, you get less migration in response to VEGF over time. And then if you overexpress endomucin, you get more migration. So at each step, the presence or absence of endomucin is modifying the ability of the cells to respond to VEGF. And then finally, tube formation, which is a sort of a lumen, obviously. People have shown that you get lumens here. It's not an ideal assay, but it has a role. If you knock down endomucin, you can see that there's many more fragments. These look less robust. And you can see that you have a reduction here. You actually have a reduction in unstimulated. So even if you don't add VEGF, you get a reduction. But this is the VEGF. And this is much more recognizable or easier to appreciate, that when you overexpress endomucin, you really get much more robust looking tubes. They just look more stable. And again, you get it in response to VEGF in both circumstances. When you overexpress endomucin without VEGF, you still see improved tube formation. So the question was, what's going on here? And this is just a pretty picture. I put co-localized in quotes here because you obviously can't co-localize something at the light microscopic level, but you can see that they sort of are in the same general area as you'd expect. So this is VEGFR2 and this is endomucin. They're both cell surface associated, as you'd expect. And it doesn't look like the presence or absence of VEGF changes that in any way. So not a ton of information from that, but does look cell surface localized. So then the real question was, what is the endomucin doing with respect to R2? And so we did IP'd with endomucin attached to beads, and then blot for VEGFR2, and then blot for endomucin. And the take home of this slide is that the endomucin does interact with VEGFR2, and it appears to be independent of whether or not VEGF is around. So it appears to be independent of whether or not VEGF is actually on VEGFR2. There's not a big significant difference here. So there's some kind of a physical interaction. We haven't quantified it in terms of affinity in any way. We haven't biocored anything. We just know that they do interact at some level. We then wanted to see what was going at the level of internalization. So who did this? Let's see, Cindy? No, Michelle did this experiment, Michelle LeBlanc. So she took these, remember, these are human retinal endothelial cells, which we know express R2, obviously. She adds an antibody to VEGFR2 to the cells. This is all done at 37 degrees, 30 minutes. Then adds VEGF. So she's first labeling the receptor and then adds VEGF so that the receptor will bind to VEGF and then start to internalize. Have pimiquid biphosphate here so we don't get recycling, because we just want to see what's happening with respect to receptor internalization here, and eventually in the presence or absence of endomucin. So then you allow that this to internalize, and then you do a low pH wash, which knocks everything off the cell surface, and you permeabilize so you can get your secondary antibody in and see what's going on inside the cell. So we're basically looking visually to see if the receptors are internalized. I'll show you some biochemistry also, though. So this is what you'd expect. These are the controls. So these are pictures of the retinal endothelium. If you add BSA, and you, again, remember you're staining for R2, you're not going to see a whole lot of VEGF-R2 internalization. It's just basically staying on the cell surface, very distributed. However, if you add VEGF, remember we're tracking where the receptor is going, you can see now that there's a lot of VEGF-R2 that's been internalized, and that's shown at a higher magnification here versus when there's no VEGF around. And this is just a quantification of that based on fluorescence. So now we're able to ask what happens with endomucin. Does endomucin influence this? And what I'll show you in the next, in the data on this slide is that when you take endomucin away by siRNA, you prevent the VEGF-R2 from internalizing. So this is what I already just showed you here. This is add VEGF without knocking down endomucin, and you have good internalization. But if you knock down endomucin, add the VEGF, there's no difference, meaning that the VEGF is added, but the receptor is not internalizing. And that's these data here. I'm going to show you more convincing. I mean, this is, in my opinion, convincing, but you want to see this done biochemically to be 100% sure, because this is a particularly interesting finding. Because what we know about VEGF signaling, and signaling from most growth factors, is once the ligand binds to the receptor, this is a tyrosine kinase, VEGF-R2 is a tyrosine kinase, gets phosphorylated. And then the receptor gets internalized by a clathrin-mediated process. But the receptor continues to signal within the cell. So the signaling happens straight from the time that it's phosphorylated as the receptor passes phosphorylated through the cell, this continued signaling. So if you don't internalize, you basically don't get signaling. So I'll show you this again. This is just, again, by pictures. Here you can see that if you add VEGF, you don't see distinct surface staining anymore because it's intracellular. You know, it's consistent with what I showed you. This is the more important piece of data. In this case, what Michelle did was to take the retinal endothelial cells and biotinylate them. So add VEGF, allow it to go for 30 minutes, then biotinylate the cell surface, and then Western blot the cell surface. So what you're asking is, and what you're expecting is, if VEGF is being VEGF-R2 is being internalized in the presence of VEGF, you will see less remaining on the cell surface. So this is how the cells start out. You add VEGF, it gets internalized as you'd expect here. But if you take away endomucin, as you are doing here, you don't internalize it. It remains stuck on the cell surface, which therefore means it's not signaling, which is why we're not getting the biology at the other end. Now, in terms of more mechanism in that area, so this is just the difference here. This shows you how much reduction in cell surface you get, and this shows you there's no reduction in cell surface. An important piece of trying to figure out what's going on here, I personally thought, you always have a preconceived notion of what you're going to see. I thought the endomucin was going to go into the cell with the VEGF-R2. It does not. So this is the same kind of experiment that I showed you before. So this is what happens when you add VEGF. This is where endomucin is on the cell surface. This is the same kind of labeling, excuse me, to go back, the same kind of labeling study that we did before, where we're labeling endomucin on the cell surface and then following it into the cell. And you can see you add VEGF and you get no internalization. And then you also get no internalization in the absence of VEGF. So endomucin is required for internalization, but is not traveling into the cell with the VEGFR2. And did that again by biotinylation. So here's immunohistochemistry, and here's the same biotinylation study that I showed you before. Endomucin on the cell surface, add VEGF, wait 30 minutes. Endomucin still on the cell surface, no difference. So I did go for 40 minutes. Now I'm gonna, I have additional data that I can show you if you want me to go into another section. This is summarizing what I've talked about so far, showing that, that endomucin regulates migration, proliferation, and tube formation in vitro. That if you interfere with it during the vascularization process, you get impaired vascularization. It does bind to R2 and appears to be necessary for clathrin. We've shown in separate studies, and it was actually known in the literature that VEGFR2 is internalized in a clathrin-mediated process. The other option would be micropinocytosis or macropinocytosis, but this is mostly clathrin-mediated. And then I have data which I have with me, but I thought maybe just for me a discussion would be more interesting. But we've done some structure function studies where Ping Zhu took the full-length endomucin and made truncation mutants of it. The truncation mutants were designed to cut off particular segments of O-glycosylation. And we showed that whereas the full length is about 160, 150, 144 amino acids, you need a minimum of 121 amino acids to get all of these functions that I've showed you. So the way she did those experiments would be she would take the retinal endothelium, knock down endogenous endomucin, and then put back in with adenoviruses the full length, that's the control. Does the full length rescue? Yes, it does. And then various truncation mutants until we got to the size that was insufficient to support rescue of VEGFR2 internalization or biology. So it's a long series of studies to show what the minimum was in that case. Other things that we're we do have mice that we made that are floxed, and we're working on those now. We just got them a little while ago. We were expecting those mice to be embryonic lethal, which is good and bad at the same time. Embryonic lethal, nice, means like that's such an essential molecule, you can't live without it. Also means, oh, you better have it floxed or you can't do anything interesting in it because it's embryonic lethal. So far, it does not appear to be embryonic lethal, which is very interesting considering the effect that it had on angiogenesis. This is talking in a direction of where we want to go with this in a more translational mode. This is a retina from a mouse model of oxygen-induced retinopathy. So it's a model where you put newly born pups with the mom in high oxygen. The high oxygen interferes with retinal vascular development, the centripetal retinal vascular development that I showed you. When you bring them back out into room air, you get a compensatory angiogenesis. It's a mouse model of a human disease called retinopathy of prematurity. And I'm simply showing you this to show you these are the fronds of new blood vessels that grow pathologically out into the vitreous, and they're very highly stained for endomucin here. We're doing just these kind of control studies. We've done also with the CMV model, and we were about to start doing siRNA here, but now we have knockout mice, so it seems not worthwhile to do siRNA, so we're gearing up to do those. In my opinion, it's possible that it's not embryonic lethal, but that a post sort of development challenge like that could still be interfered with by interfering with endomucin. And the reason that we're interested in this whole, let's see if I put it in my next slide, I'm sorry. So this is just a general conclusion, which is the glycocalyx has got a lot of really interesting things to study. very understudied if you look at the literature in terms of the kinds of studies that are out there. Translationally, what we're interested in here is, as you well know probably, VEGF's a important target for things like diabetic macular edema, wet macular degeneration, proliferative diabetic retinopathy, et cetera. We're thinking that this may be a better target because it's endothelial cell specific. So VEGFR2 is actually pretty ubiquitous, even though it's called vascular endothelial growth factor receptor, it's actually much more widely expressed. It's found on photoreceptors and Mueller cells and astrocytes and RPE cells. And therefore, when you block VEGF by sequestering the ligand, which is the clinical way to do it right now with aflibercept or Lucentis or Avastin, you're blocking VEGF that is needed by some of those cells. So VEGF's a survival factor for RPE cells. You're blocking all of that. So if you could block VEGF specifically at the endothelial cell level by blocking endomucin, because it's only expressed by the endothelium, you may have a more cell-specific inhibitor. And that's what we're thinking about testing at this point once we get the assays going. These are the people who have been doing the work. This is what your lab meeting looks like, I'm sure, these days. Cindy Park started the project. Well, actually, Eric started it many years ago by finding the endomucin. Cindy did the retina stuff in the early proliferation migration assay with Magali. Michelle did some of the mechanistic work, and Ping now is working on looking at internalization studies, like where in the internalization process are things going wrong when endomucin is not there. And Dimar Vestweber gave us some anti-sera, which we've been using because it became not available anymore commercially, and that was not good for us. And no, that's the truncation mutant data. I won't go into that. So I'm happy to entertain questions, stop here and entertain questions. Suggestions in particular would be welcome. Ideas about directions to go, any endocytosis internalization experts online. Love to hear what you have to say.
Thanks, Pat. That was elegant and very interesting. I'm going to ask if people have questions. I can't see you all. So I'm going to ask you to open up the chat box and just put your name in the chat. You don't have to type out your whole question if you don't want to, but that'll just let me know that I should call on you. So it looks like Jorge Alvarez has a question, so you could unmute yourself and ask it.
Thank you for your talk. This aspect of endothelial biology is something that it has always been of my interest, and it's nice to see that somebody is talking about it. What I'm curious to know is how endomucin expression is regulated, and given that you show it during development, is the expression the same during development versus in other stages of the organism development, like adulthood?
Yeah, good question. We don't know a ton about expression. We obviously know that it's associated with VEGF at some point, because when we knocked out VEGF, we got reduced endomucin. And if you add VEGF back to those embryoid bodies, you can get elevated endomucin expression. There's also one paper in the literature that shows it's downstream of GATA4, because it is expressed very early on in hematopoietic development. Not later on that I know of, but earlier on in hematopoietic development. In the retina, for instance, the levels go down in the adult. And I think this is not backed by a lot of data, it's backed more by observation. The levels are going to correlate with R2 levels. So if you look at choroid, which is a fenestrated vascular bed, which has a lot of VEGFR2, there's a ton of endomucin there. If you look at the retinal vasculature in the adult, R2 is very downregulated there because those cells are extremely quiescent and endomucin levels are low there. but that's a observation at this point. I haven't done anything, systematic to look at it, but I'm getting the feeling that it's correlated with R2 expression.
So from what you alluded, it seems that it plays a role in regulating the migration of immune cells, which makes me think that it kind of like promotes some sort of immunoquiescent state on endothelium, but it's kind of like doesn't go with that hypothesis if you say that there is more in the fenestrated endothelium versus the blood retinal barrier. Is that a fair assessment?
Sentence summary of the role in inflammation. If you take resting cells, and for those we mostly used umbilical vein cells because people use umbilical vein in that field when you're looking at leukocyte adhesion. And you do one of those flow assays where you take neutrophils and you flow them across the surface of the umbilical vein. You don't get sticking normally unless you've thrown TNF on there or something. If you knock down the endomucin, you get way more sticking. And when we looked a little bit more deeply, it turns out that when you take away the endomucin, you're revealing on the surface of the endothelium constitutively expressed ICAM. So there's ICAM there all the time, not a ton, but enough to mediate adhesion. And then if you come along on top of that and add a pro-inflammatory like IL-1 beta or interferon or something, you get rid of, independent obviously of the SI, you get rid of the endomucin, it gets cleaved proteolytically, and then all the adhesion molecules come up. So as part of inflammation, I think sort of clearing away the brush of the glycocalyx, which is what people have shown in sepsis, is that when people are septic, the glycocalyx is greatly reduced. The idea is that you're cleaving probably not just this glycocalyx molecule, but others to make the cell surface of the endothelium and all the adhesion molecules sort of maximally accessible to the leukocytes as they go by. So having it there normally, so you always have some ICAM there, which I wasn't really aware of, but there's low levels. And that makes sense because if you have inflammation, you don't want to have to wait until you like make ICAM or the waiting. So, so in that regard, it is sort of an anti-inflammatory function, frankly, in the same way that the endothelium is anticoagulant because it has heparin sulfate on the cell surface and makes some prostacyclins. So it has that sort of low level, sort of homeostatic, trying to keep the endothelium quiescent.
Thank you.
So can you finish your thought though about what you thought didn't connect between the,
because like the blood retinal barrier, for example, is more immunoquiescent than the fenestrated endothelium. So like
yeah,
if the
true, but they also have the blood-brain barrier has the many other backup. I wouldn't say the glycocalyx is the prime site of the sort of immune privilege of the blood-brain barrier. It's you don't have any lymphatics, you have really tight junctions, you don't have any transcytosis. There's a specialized receptors are very down-regulated. So it's just a small, I think there's so many other aspects of the blood-brain barrier that are at play that the fact that endomucin might be a little lower is not going to change things, in my opinion.
Yeah, but I was talking about the blood retinal barrier.
Same. Mm-hmm. Blood retinal barrier is the same. It's got low transcytosis, tight junctions, no lymphatics. So all the things that make the blood-brain barrier, a blood-brain barrier and blood retinal barrier, unless I'm wrong, are pretty similar. Do you know of big differences between them?
I was more focusing on, like, for example, these fenestrated vessels that are close to the retinal barrier and the retinal barrier, right? For the migration.
Yeah, but those, the fenestrated vessels aren't that close. They are separated by RPE, which is the outer blood retinal barrier. That's a very tight junction to get across. So that has very high resistance in a way that blood retinal barrier has. So even though the choroid is pretty close in distance, it's still very protected by the RPE barrier.
Thank you.
Yeah.
But I have a question. So does endomucin potentiate VEGF binding to VEGF receptor 2 or the endocytosis of the receptor once VEGF has bound? And I guess you might know that if you determined whether the receptor is phosphorylated in the presence of VEGF, but the absence of endomucin.
It is. So when you knock down endomucin, you still get receptor phosphorylation. And, you know, you can fool around with that in terms of nuance by looking at different phosphorylation sites, but it's not significantly blocked. So it really is about the internalization part of it. And that's sort of where our efforts are now, like trying to understand, because that whole area of internalization have a lot of, I mean, the way I'm thinking about it now is that the endomucin, even though we haven't found out anything that the intracellular domain is doing, I can only imagine that there must be something, we haven't been able to catch it phosphorylated, if you will, even though there's like 6 phosphorylation sites and they're all really conserved. We haven't been able to document it being phosphorylated, but I'm imagining that it may be some kind of a adapter that's bringing in things like epsins that we know you need for receptor internalization. That's sort of where we're at in that project right now.
All right. Everybody else, please type in that you have a question. I've only seen one from Jorge so far. So I'll ask another one. We can go for about another 7 minutes. So what happens to endomucin in disease that, how is it modified that might promote neovascularization? I think you mentioned that you saw some changes in it with disease.
Yeah, I mean, that's a really good question, trying to merge all of the knowledge that we're getting. So in acute inflammation, when you have something like IL-1 beta or, you know, TNF, the extracellular domain gets cleaved off. I mean, it's really, you know, within 6 hours, it's gone. And the trouble with trying to, what we haven't done, which somebody's actually trying to do right now, is see when you add TNF like that, if you follow up with VEGF right away, will you get VEGF signaling? Will you still get, but I suspect you'll, yeah, actually we don't have the answer to that. I really don't know what the answer to that's going to be. It's when you try to merge these somewhat distinct functions of the molecule, you know, like mediating or allowing internalization with what's going on if it gets totally clipped away in the course of inflammation. We just really haven't figured that out yet. I don't know. I'd be interested if anybody had a suggestion. The thought would be that once it's clipped, that VEGF is not going to be stimulatory anymore. And I don't know if people have really looked to see, can you be activated? In other words, inflammatorily activated as an endothelium and proliferate at the same time? Or are those mutually exclusive phenotypes? My sense is they might be mutually exclusive, and then you might have locally different phenotypes. So, you know, this subset are going to be expressing adhesion molecules and doing what they're doing, and then maybe some cells down the line are going to undergo angiogenesis. It's a hand waving response. I don't really know. But, you know, angiogenesis is associated with inflammation. So at some level, you need to have it there. And we haven't figured that out yet. Or if there's even maybe a backup molecule. In culture, there isn't. But I mean, the fact that the mice are not embryonic lethal would suggest that there might be some redundancy and there's something else that can do the same thing. So that's another project to figure out.
All right, thank you. So Claire has a question. Why don't you go ahead and unmute yourself and ask Pat?
Oh, altered with age or gender. I'm reading it. Do you know whether they're altered with age or gender? I do not know either of those things. No, I don't know. I don't know if there's gender. I don't know if there's any sex hormone regulation of it. And she says her mice are not working. I think she means her mic are not working. That was a good Freudian slip. But age-wise, you know, again, a hand waving response is that everything declines with age. I hate to say it, but I wouldn't be surprised if there are changes, and it could even be something as
Well, it looks like we lost her again in the middle of that. So
in glycosylation at this point. The age
hey,
part.
Pat, could you back up about 30 seconds? We lost you.
Oh, geez, really?
Yeah.
Well, I didn't say anything that useful because I said we have no idea about gender at this point. I wouldn't be surprised if there are age-dependent effects, because there are age-dependent effects in everything, but we have not documented it. And if it's not levels, it could be glycosylation pattern, which is much harder to study, to like sequence glycosylation pattern. It's possible, but it's not someplace we've gone. But it would be interesting since, you know, vascular density declines with age. What is that a function of? Is it a function of VEGF, the receptor, the endomucin? Don't know, but those are good questions.
One more question. So to follow up on what you said about inflammation in endomucin, how does IL-1 beta and TNF upregulation result in cleavage of endomucin? Do you know?
Yeah.
Is there a protease or a cell?
Yeah, they're MMPs. So we've looked at the various MMPs, and it leads to cleavage at the cell surface. We're not exactly sure of the site yet. We're trying to figure out the site. Those. It turns out that those are the same MMPs that are not surprisingly involved in cleaving A levels also later on. So it acts at both levels, but quickly, you know, within 4 hours, you have a great reduction in cell surface just clipped off. Interestingly, though, it's hard to find the clipped off part. I don't know if it gets chewed up right away. You can have a big bunch of cells, clip it off, and then try to collect the conditioned media to get the cleaved fragment and not find it. So that's very bizarre, but it definitely gets clipped.
Sorry, we lost you in the middle there. Are the endothelial cells making the MMPs in response to the cytokines?
Yes, yeah, because it's just a single cell system there. So it's definitely the endothelium, yeah.
All right.
Sorry about my internet being so unstable. It says it's
it really
good, but
wasn't a big problem, just a couple of times, and
okay.
you got frozen. Yeah, maybe it's freezing up there in Boston, you know.
It actually is freezing out in Boston here. It's like 20, so I guess it affects the internet too.
Yeah, probably. All right, well, thank you again so much for joining us, Pat. That was
all right, thanks everyone.
Great.
And I had said, I didn't plan meetings with anybody today because it's Zoom, you can meet them with anytime. If anybody really wants to